Hi friends, today I am discussing about the topic is immunology. Immunology is the first we have to know the definition of immunology. Immunology is the study of immunity is known as immunology. Immunology is the science that is immune response to foreign challenges. Immunity derived from Latin word. Immunity is ability to resist infections by pathogens or protection against foreign organisms. This immunity carry of cells, organs, tissues. This activity is known as immune system. This immunity is divided into two types: innate immunity. Another one is the adaptive immunity. Innate immunity is the natural immunity. It is present since birth. Innate immunity is the natural immunity. It is present since birth. It operates against almost any foreign molecules. It operates against almost any foreign molecules and pathogens. It provides first line of defense against pathogens. It is not specific to any one pathogen, acts against all foreign molecules and pathogens. Next one is the elements of innate immunity. Innate immunity elements, first is the physical barriers. Physical barriers are the first line of defense against microorganisms. It includes skin and mucous membrane. Most of the organisms and foreign substances cannot penetrate intact skin but can enter the body if the skin is damaged. Another one is acidic pH of sweat and sebaceous secretions and presence of various fatty acids and hydrolytic enzymes like lysozyme inhibit the growth of microorganisms. Similarly, respiratory and gastrointestinal tracts are lined by mucous membranes. This is entrap foreign microorganisms. Tears contain lysozyme, lactoferrin, IgA antibody and provide chemical as well as physical protection. This innate and adaptive immunity systems to recognize the foreign pathogens and destroy them without harming the host. Innate immune response, several antimicrobial chemicals and phagocytic cells provide protection against pathogens. And next one is the chemical mediator. First one is the physical barriers and second one is the chemical mediator. Chemical mediators are complement proteins, cytokines, pattern recognition molecules, acute phase proteins, cationic peptides, enzymes like lysozyme and many others. This first one is the complement proteins. These complement proteins are soluble proteins or glycoproteins that are mainly synthesized by liver and circulate in the blood and extracellular fluid. They were originally identified by their ability to amplify the complement, the action of antibodies, hence the name complement. This complement system composed of over 30 serum proteins, activation of complement proteins in response to certain microorganisms results controlled enzymatic cascade which targets membrane of pathogenic organisms and leads to their destruction. And next one is the cytokines. Cytokines is also elements of innate immunity and cytokines is generic term for any low molecular weight of soluble protein and glycoprotein released by one cell population. Act as intercellular mediator. It includes monokines, lymphokines, interleukins, interferons and others. Cytokines are required for immunoregulation of both innate as well as acquired immunity responses. Interferons are cytokines made by cells in response to virus infection, which essentially induce a antiviral state in surrounding cells. And next one is the chemokines. Chemokines are small positively charged secreted proteins that have a central role in guiding the migrations of various types of white blood cells. They bind to surface of endothelial cells and to negatively charged proteoglycans of the extracellular matrix in organs. By binding G protein linked receptors on the surface of specific blood cells, chemokines attract the cells from bloodstream into an organ organ, guide them to specific locations within the organ, then help to stop migration. And fifth one is the pattern recognition molecule. Many molecules involved in innate immunity have the ability to recognize a given class of molecules, that is recognize pattern. In this molecule, In this molecule recognize pathogen-associated molecular pattern may be soluble circulating protein cell surface receptors. Many pathogen-associated molecular patterns are recognized by pattern recognition molecules present on the surface of phagocytic cells. And another one is cellular defenses. Cellular defenses like neutrophils, macrophages, natural killer cells participate in innate immunity host defense mechanism. Once pathogen evades the physical and chemical barriers, the specialized cells play an important role in protection. Phagocytosis is a fundamental protective mechanism carried out by neutrophils, macrophages, monocytes, and dendritic cells. In vertebrates, macrophages reside in tissue throughout the body. Macrophages are long lived cells which patrol the tissues of the body. And the second major type of phagocytic cells in invertebrates, the neutrophils are short-lived cells which present in blood but are not present in normal healthy tissue. Phagocytosis is ingestion of invading foreign particles such as bacteria by individual cells. Phagocytosis may be enhanced by a variety of factors called opsonins. Greek word meaning prepared food for, which consist of various serum components and antibodies of complement. The phagocytosis process by which particulate antigens are more susceptible to this process called opsonization. After ingestion, foreign particles is entrapped in a phagocytic vacuole, that is phagosome, which diffuses with lysosomes forming phagolysosome. This phagocytosis is this phagocytosis mechanism is two types: oxygen dependent killing mechanism and oxygen independent killing mechanism. Now, oxygen dependent killing mechanism is a metabolic process known as respiratory burst. Occurs in activated phagocytes. It results in a transient increase in oxygen consumption by cells. Activated phagocytes generate a number of toxic products such as reactive oxygen intermediates. That is, oxygen intermediates are hydroxyl radicals, hypochlorite anion, superoxide anions, and hydrogen peroxide. And also reactive nitrogen intermediates and toxic products, oxygen intermediates, and also nitrogen intermediates, that is nitrogen monoxide, nitrogen dioxide, nitric acid, which have potent antimicrobial activity. And next one is the O2 dependent killing mechanism. Activated macrophages also synthesize lysozyme defensins. Defensins is a cysteine-rich cationic peptides containing 29 to 35 amino acid residues and various hydrolytic enzymes, cytotoxic peptides whose degradative activities do not require oxygen. And next one is the inflammatory barriers. Inflammation is an important non-specific defense reaction to cell injury. The hallmark signs of inflammation are pain, redness, Redness means erythema, swelling means edema, and heat. Each of these results in specific changes in local blood cells. Erythema is caused by increased vascular diameter, which leads to increased blood flow, thereby causing heat and redness in the area. The inflammatory response is mediated by a variety of signaling molecules. Activated macrophages produce chemoattractants called chemokines. Some of the attracts neutrophils, which are the first cells recruited in large numbers to the site of new infection. Others later attract monocytes and dendritic cells. First attract neutrophils and other attract monocytes and dendritic cells. Two principal mediators of the inflammatory response are histamine. This histamine released by a variety of cells in response to tissue injury. And second one is the first one is the histamine and second one is the kinins. Kinins present in blood plasma in a inactive form both cause vasodilation and increase permeability of capillaries. Kinins are also very potent nerve stimulators, are most responsible for pain associated with inflammation. First one is the innate immunity and second one is the adaptive immunity. Adaptive immunity is also known as specific or acquired immunity, is capable of recognizing and selectively eliminating specific foreign antigens. In this adaptive immunity displays four characteristics. Antigenic specificity is the ability to discriminate among different epitopes and antigens. And second one is the immunologic memory. It is ability to recall previous contact with a foreign molecule and respond to it in a learned manner, that is more rapid and larger response. And third one is the diversity, the ability to respond to different epitopes. And fourth one is the self and non-self recognition. It is the ability to recognize and respond to molecules that are foreign and to avoid making a response to those molecules that are self-tolerance. The differences between the innate immune response and adaptive immune response. Specificity of the innate immune response is antigenic non-specific. Specificity of the adaptive immune response is antigenic specific. Response time in innate immunity is rapid response hours. Response time in adaptive immunity is slow response days. Diversity limited. Diversity in innate immune response is limited. Diversity in adaptive immune response is very high. Memory of the innate immune response is absent or low. Memory of the adaptive immune response is present. Major cell types in innate immune response: phagocytes, natural killer cells, and others. Major cell types in adaptive immune response: T and B cells, antigen presenting cells. Distribution in innate immune response found in vertebrates and invertebrates. Distribution in adaptive immune response found only in jacked vertebrates. Adaptive immunity may be active or passive. Active immunity induced by natural exposure to a pathogen and or by vaccination is the artificial. In this kind of immunity, a person develops own immune response to a microbe. Immunity resulting from the transfer of antibodies or immune cells from an immune to non-immune individual is known as passive immunity. Passive immunity means transfer of antibodies or immune cells from an immune to non-immune individuals. This is passive immunity. It may be naturally involves the transfer of antibodies from one host to another or artificially when antibodies or lymphocytes that have been produced outside the host are introduced into host acquired immunity. And next one is the differences between active immunity and passive immunity. This active immunity is the exposure to antigen and passive immunity is the no exposure to antigen. Immunity achieved by injecting antigens in active immunity. Immunity achieved by injecting antibodies or antigen reactive T cells in passive immunity. Activation of immune system in active immunity. No activation of immune system in passive immunity. Immune state develops over a period of weeks in active immunity. Immune state develops immediately in passive immunity. Immunological memory develops in active immunity. No immunological memory is present in passive immunity. And next one is the immune response of adaptive immunity. Two arms of acquired or adaptive immunity, different sets of participants and different purposes, but one common aim to eliminate antigen. Two arms interact with each other and collaborate to achieve the final goal of eliminating the antigen. Two arms of acquired immune response. One is mediated mainly B cells and circulating antibodies, a form of immune